For a compare and contrast question, it really means find the similarities, which is comparing, and find the differences, which is contrast. So this types of questions, for me, I'd like to make a table format and kind of compare and contrast between. And if we are given different types of species or something like that, I would make them into a different column. So here we go. Compare and contrast the cell wall and cell membrane components of gram-negative and gram-positive. So here, this is a table I made. On the left is a column containing all the components that I would like to make notes about. So gram-negative and gram-positive have each own column. So if we start with peptidoglycan, well, it's found in both. Make sure you put it's found in both, as that will get you some marks. I didn't, but you know the point. So gram-negative is actually monolayer. They have very thin peptidoglycan. And as you see on the next point, the reason gram-negative have thin peptidoglycan is because it has another membrane. And this difference, well, now what's the difference here? Gram-positive have commonly tri-layer peptidoglycan, or it's considered multi-layer. And also they have teichoic acids that are embedded in the peptidoglycan. So now you can see the thickness is different, and this is what allows us to be able to perform gram staining using crystal violet and safranin as the major or main dye. And the second point is outer membrane. Well, in gram-negative, outer membrane is present, and this outer membrane is linked to peptidoglycan by lipoproteins. Outer membrane contains porin, so porin, which allows hydrophilic molecules to be able to move across the membrane in and out more freely. And also, our outer membrane contains lipopolysaccharide, more commonly known as LPS, which extend into the intracellular space. But in gram-positive, they don't have outer membrane, so all they have is cytoplasmic membrane or plasma membrane and peptidoglycan, and that's it for them. Gram-negative, they would have plasma membrane, periplasmic space, and within periplasmic space there is peptidoglycan, and they would have another membrane called outer membrane. So here's another difference here. Another one is that in gram-positive they contain periplasmic space. So peptidoglycan is actually found here. However, in gram-positive they don't have periplasmic space. The last point is gram-negative and gram-positive both have plasma membrane. But the difference here is in gram-positive, it also contains lipoteichoic acid, which is extending from peptidoglycan all the way outside of peptidoglycan, all the way into the cytoplasmic or plasma membrane. So this is their main difference and similarities.